and they give an academic talk and then this awesome public lecture. And so today I'm going to be introducing Ian Wishaw, who will be introducing our guest of honor. So Dr. Wishaw is a prof in neuroscience at the U of L. He's been there forever. He's done some awesome things. He has over 300 publications. I think you're top cited in Canada, top 200, yeah, something like that. And he's also very good at riding horses. So without further ado, this is Ian Wishaw.
Thank you, Candace. As Candace said, my name is Ian Wishaw. Why am I up here? What do you want me to do? I'm going to tell you a story. My colleague Brian Cove isn't here. Many of you may know him. He's also a professor at the University of Lethbridge. But when he graduated from university and then did a postdoc, he started applying for jobs. And he applied to about 100 places all over the world, and they all turned him down. And for a while, he took these rejection letters and he pasted them up around his office. But when he finally decided to take them all down, and he's got them in a big pile, and he keeps them in his desk in a top drawer. He shows them to students, and I don't know why he does that, but he does. Anyway, when he couldn't get a job, he got a postdoc at McGill University to work with Brenda Milner. Now, postdoc isn't a job. It's just what you do when you can't get a job. And from Brenda Milner, he learned neuropsychology, which is the study, really the study of how the brain produces our behavior, produces our memories, produces our cognitive processes, and all of the kinds of things that the brain does. And armed with that information, Lethbridge hired him. And when he came here, I said, well, you've got to teach me neuropsychology. So he taught me neuropsychology, and we wrote the first textbook on neuropsychology, which still comes out. And I'm telling you this story is because this is how we know Brenda Milner, through Brian. And she's been a good friend to the university all these many years. When she first saw our book, she said, I can't believe neither of you can write. But that's the way she is. And she got an honorary degree here. And when the Brenda Milner lecture series began, it was really the students that began it. And they wanted to bring in a speaker of their own, the kind of people whose papers they read when they're doing their theses. And they asked for some money to sponsor a speaker. And whenever the university has a budget crisis, the first thing that goes is the speaker's budget. So we've never really ever had a speaker's budget because we've had many budget crises over the years. So the students were told, well, you have to go and raise money for yourself. And so they did. They went out and raised the money, and they've been doing that every year and bringing the speakers in part with donations that come from people. And one of my jobs tonight is just simply thank the following people for the donations they've made. Naomi Kramer, Ian Wishaw, that's me, Brian Cove, Masami Tazama, David Houston, Robin Gibb, Majid Mohidjahir, Robert Sutherland, and Cheryl Aurelius, and David Carpenter, and Gen Manufacturing, the President's Office, the Alumni Office, the Society for Graduate Students, the Education Faculty, and the Brain Diseases and Department of Neurosciences. So I want to thank all of those people who made donations. I know the students have worked very hard, and I want to thank them for the work that they've done as well in organizing this. It's taken them nearly a year to put all this together, and I know during the course of it, they told me that our guest speaker today, Eleanor McGuire, whose name is up there, only gives 2 talks a year. That's not true. Yes, it's a good story. And she's giving those 2 talks here. So our speaker, Eleanor McGuire, I think will give you a fascinating talk. She's from Dublin, Ireland. She got her undergraduate degree from University College in Dublin. Then she went to the University of Wales where she got a master's degree. And then she went back to Dublin and got her PhD. And since then she's been at the University of London. She's a Wellcome Trust fellow. And I think with that, I just want to say one more thing, if you permit me. I think I read somewhere, you said to be a good scientist, you have to have two things. You have to have a theory, and you have to be able to give a talk. And I know you have a fabulous theory, and I know you're going to give us a fabulous talk. So it's all yours.
Thank you. No pressure then. Thank you very much. Well, I'm absolutely delighted to be here in Lethbridge, and thank you all for coming. And again, I want to thank the committee for all their hard work in making this happen. So I spend my time trying to understand how memory works and how the brain supports this crucial function. And I say crucial because memory impinges on everything we do as individuals and as a society. So for example, you have a sense of who you are, you have an autobiography, you can live independently because of memory. And we share beliefs, we share laws and culture because of memory. So it's really important, and of course that's why we fear losing our memories. Now, here's a kind of spoiler for you, we still don't really know how memory works. But what I'm going to do is tell you how we go about studying memory, tell you what we used to think about memory, what we think we know now, and where I think we might be going in the future in our pursuit of the memory code. So the first thing to say about memory, of course, is that it's not one thing. There are many different types of memory. So for example, you have short-term memory. These are things like phone numbers, and I mean, I'm going to have to change this slide because most of this now is your mobile phone, right? So I'm not sure we use our short-term memories as much as we used to. But this is for just short-term information. And then we have longer-term memory, and there's different types of memory. There are things like riding a bicycle. You don't have to think about it. It's kind of procedural. And then there are these types of memories. So the ones that we're particularly interested in are here. Sorry. These are episodic, or we call them autobiographical memories. So these are our memories of our past experiences. And then there's kind of memories for facts about the world, concepts, the vocabulary that we acquire through our lifetime. So there are many different types of memory. But the one thing to say about memory is that it is not a perfect record of the past. Memory is very much fallible, but it's also flexible, and that serves us well, as you'll see. So, how do we examine memory? Well, one way we do it is we test people's memories with tests using paper, pencil, computerized tests. And we can examine memory, and one thing we're interested in trying to understand is why in a group of healthy people do some people have good memories, some people have bad memories. So what is the basis of individual differences in memory? And so that's one way we study this. These type of data also serve as a comparison to make to patients. Sorry, this is very...
Yeah.
Okay.
Might be your boss.
Oh, I can see without my glasses.
Do you want to try the headphones?
Yeah.
Let's do it. Sorry.
Okay, that's much better. Yeah, thank you. So we also test patients, as we heard from Ian, neuropsychological testing. So we test people's memories, other aspects of cognition, and people can have different types of brain damage. It can be a very focal damage like here, or it can be something more diffuse like dementia. Now these types of scans are magnetic resonance imaging scans, or MRI scans. And this is another technique that we use to study memory. And some of you might have had an MRI scan over the years. It's like going into a tunnel, a very noisy tunnel. But what we get are amazing pictures of people's brains. For example, we can get pictures, 3D pictures of people's brain, and we can see the grey matter, the white matter. Here's some views. This is the front, facing front of the brain, side view, a view top down. And you can see the amazing detail that we can examine in the structure of the brain. And we can also look at the connections between brain areas. And this is actual what it looks like. I mean, it's not this colour, obviously, but it's this intricate. So there's all this connectivity, and we can start as well to try and correlate different aspects of brain structure with memory functioning. So that's one way we can use these types of data. Now we can also study function directly using MRI. This is known as functional MRI. And what you're seeing here are different parts of the brain that are active when somebody is doing a task in the scanner. And this works very sort of simply speaking by when a brain area becomes active, the blood flow increases to that brain area to sort of feed the neurons that are working hard. And we can measure those changes in blood flow as a sort of indirect measure of neural activity. And that's what we're looking at here. Now the thing about this blood flow is that it's often quite slow. So there's about a 6-second lag between something happening and us being able to measure the blood flow. So although fMRI is very good about telling you where changes are happening, it's not so good about telling you when changes are happening. So we need to look for a different technique for that. And so we use things like electroencephalography, or EEG, or in this case here, magnetoencephalography, or MEG. And this is like one of those old-fashioned sort of hairdryer type things you'd see. And somebody sits there and they do a task, a memory task. This is the screen. And then we can look at their neural, at the output of their neurons directly while they're doing a task. And the spatial resolution, we can tell roughly where we are in the brain with this, but we have millisecond precision here. So the temporal resolution is amazing. Now the next slide, if you're of a squeamish disposition, you should look away now. We can also study electrical activity from the brain, or neural activity, directly. So these are patients that have intractable epilepsy. And the idea is that we want to find out where the source of their seizures is coming from, and hopefully we can try to remove that tissue so we can reduce or eliminate their seizures. But it's not always easy to tell where the seizures are coming from, so we put all these electrodes directly on the brain, in this case, or into the brain, deep into the brain. We put these depth electrodes in. And so we can record not only the seizure activity when it's happening, but we can also get these patients to do different types of memory tasks. And then we can measure their brain activity when they're doing that. And the same principle works for animal testing. So this is another technique we use. So we can have rats in a maze doing spatial memory tasks, but we can also measure electrical activity or neural activity directly from the brains of the animals while they're doing the tasks. Now the thing to say about all these different techniques is that no technique is perfect. They all bring something to the table. And the strength, what we're looking for, is to look for convergent evidence across these different techniques. So going back then to the different types of memory, as I alluded to, I'm quite interested in this type of memory. So these are the memories of our past experiences, and that's what I'm going to focus on here initially. So these are known as autobiographical memories, and they are the sorts of events in our lives that we remember. Baby's first haircut, when my nephew met Yoda on Oxford Street in London, all very memorable events. Now we know that many brain areas support these types of memories. They're very rich memories. They involve thinking about people, places, what happened, and so on.
But there is one particular brain area that when it's damaged, we can't replicate it in the brain. No other brain area can take up the slack when this brain area is damaged. And this is the hippocampus. So you can see it here. It's kind of deep in the temporal lobes. We've one on each side. And here it is on an MRI scan in the sort of side view, the front view, and looking down. And it's called hippocampus because this is one taken out of the brain, and you can see that it does, to the early anatomists, and indeed to us, it does look quite like a seahorse. And so that's where the name hippocampus comes from. And we can see it activated here during a memory task in the fMRI scanner. And we can look at patients. Here is the hippocampi of patients, and you can see they're quite shriveled compared to these nice, juicy, healthy hippocampi in the healthy control subjects. So we do a lot of testing of patients with these types of lesions, and these patients are amnesic. They can't remember what has happened to them. Now, the idea that the hippocampus is important for memory came about really back in the 1950s with Brenda Milner. And she found this patient, HM, or Henry Molaison, who had intractable epilepsy. And what they did at that point in time was removed both of his hippocampi to try and alleviate the seizures, and quite a lot of the surrounding tissue also. Now the problem with that is they didn't realise at the time that if you take out both hippocampi, you're going to make someone completely and permanently amnesic. And that's what happened to HM. So this is a description of an interaction by Sue Corpin, who I think was Brenda's PhD student. So what do you do during a typical day? See, that's tough. What I don't remember things. Do you remember what you did yesterday? No. This morning? No. What you had for lunch? No. What you'll do tomorrow? Whatever is beneficial. So he has really got very little memory of his past experiences, and he's unable to acquire new memories. And it was on the basis of HM that people really started to understand that the hippocampus is very important for memory. So now even today, when we test patients with damage just in the hippocampus bilaterally, we often see patients like this. So here's controls recalling memories passed through their lifetime from when they were 11 to their sort of teenage years and so on. The patients typically in our hands don't remember anything that's happened to them. So the hippocampal damage is massively deleterious for their memory, and because of this, the hippocampus has become synonymous with memory. And hippocampus is very vulnerable to a range of pathology. We've already heard about epilepsy, but of course it's also very susceptible to things like dementia. So it's very important for us to try and understand what the hippocampus is doing. And here it is, as I say, in the fMRI scanner with a network of brain areas, but we're going to focus here on the hippocampus. Now, in recent years, what we've been doing is we've been able to get people to remember their past experiences during fMRI scanning, and we've been able to detect the specific patterns of brain activity associated with specific memories from activity in the hippocampus. So we can tell which memory a person is recalling just by looking at the patterns of fMRI activity. So we can tell what they're actually remembering. And so we can see how these kind of memory traces, if you like, are in the hippocampus. Here, these blue areas are supporting memories that are very recent, just formed 2 weeks ago. The red is showing memories that were much more remote, from 10 years ago. And then we can bring people back, like 2 years later. These are the same memories. This is the memories when it was 2 weeks old. This is the memory when it's now 2 years old. So we're able to track the memory traces over time and see what happens to them in the brain, and then hopefully see in patients where the memories go awry, at what point in the process. But there's an issue. So the issue was that while Brenda Milner and lots of other people were studying things like autobiographical memory and making the case that the hippocampus is really important for those kinds of memories, in the animal world, the hippocampus was being studied in relation to spatial memory and navigation. And the case there was being made by people like John O'Keefe, who recorded activity in the hippocampus and found that there were these cells called place cells that only fired when the animal was in a particular place. And it didn't matter where the animal was looking. This neuron seemed to recognize the place, so that sounds quite like a memory type cell. So let me try and play this for you. Hopefully it will work. That's the neuron firing. The rat is much speeded up than he would be normally. But you can see that the cell is only firing when he's here in this part of the arena. Doesn't matter where the rat's looking. And you can see it's silent in all the other parts of the arena. So it kind of seems to be a memory. Oh, he's really speeded up now, don't worry. It seems to be kind of a memory for that part of space. And in fact, John O'Keefe won the Nobel Prize a number of years ago for this work. Okay. Now, another thing, and some of this work is going on in Lethbridge as well, really interesting work, is that the hippocampus and space have also been linked by looking at the volume of the hippocampus. So, for example, in some breeds, here I'm talking about cowbirds, their volume of their hippocampus changes seasonally depending on the type of activity they're involved in. So, for example, during the mating season, males roaming around looking for females, the volume of their hippocampus changes. So then I wondered, based on this kind of work, whether the same thing happened in humans. And so I was searching around for a sort of population of people we could study. And what we studied was licensed London taxi drivers. So taxi drivers in London have to study for approximately 4 years, and they have to learn about 25,000 streets, how they all relate to each other, all of the landmarks that are on these streets. It's a phenomenal act of memory. So, and of course, it's the perfect navigation study to do. So we looked at their brain structure, and lo and behold, we found that there was increased grey matter in the sort of mid to back part of the hippocampus compared to controls who weren't taxi drivers. And interestingly, the front part of their hippocampus decreased in grey matter. So a lot of people forget that this is a story of gain, but also loss. So this was amazing. This seemed to kind of mirror what you find in the animals. Now this was a cross-sectional study, and people made various points about the control group. So what we did then was we compared London taxi drivers to London bus drivers. So they're all navigating in London, they're all having to deal with stressful customers and traffic fumes and everything else. But yet again we found that the taxi drivers still showed this increase in the back of their hippocampus and decrease in the front. And the bus drivers, of course, just navigate around very restricted routes, but the taxi drivers, of course, have 25,000 streets in their head. But the real test we had to do, of course, was a longitudinal study. So what we did was we got trainee taxi drivers just before they started their acquisition of the knowledge, as it's called. And we scanned their brains and we tested their memories. And then we came back 4 years later. And for those, so we had about a 50% pass rate. It's very difficult to qualify as a taxi driver. It's really tough. And there's about 50% who pass and 50% who fail. And we also had a control group who never did any training. And what you find is that it's only in the qualified trainees, this is when they started, and this is when they qualified, you see these changes in the hippocampus. You don't see it for the people who failed, and you don't see it for the people who were never engaged in any kind of taxi driving at all. And so this is the kind of evidence that it's definitely kind of plasticity in the hippocampus. And the original taxi driver study, at the time, people were not sure that the kind of adult human brain could really change in relation to environmental stimulation. But this really shows that you can. These are all sort of average IQ people, all adults, and yet their brains are able to change. Now, we also wanted to study actual navigation. But here we run into a problem, because this is the real world. This is Oxford Street in London. And this is our brain scanner, where your head is immobilized and you're not allowed to really move very much at all. How do we study navigation by, using MRI? So luckily, we can use virtual reality. And very happily for us, at this time, Sony, the video game maker, had produced a video game called The Getaway. And the reason that was interesting is because they went out and simulated the whole of central London really accurately for this video game. So we were able to take this game, and all my researchers spend their time playing these games so that we could understand how they worked. It's a kind of a gangster type video game. And so occasionally these gangsters would appear and try to mug you and things like that. So we tried to get rid of as much of that as possible and then just use it as a navigation. You can drive around the city, happily in a London taxi if you like. And so that's what we did. So people drove around London, finding their way while we scanned their brains. And so I'm just going to show you an example of what this is like. So you'll see here the person navigating. This is a map of London, and you'll see them moving where they're going. Here you'll see the different brain areas that are active, and you'll see here the thoughts that these people were having at exactly the time they were navigating. So I'm going to play this movie, and you have to watch out really quickly at the very start. Around this point here, you'll see the hippocampus come on. And that's when people were planning their routes. So we would say, go to X place or Y place. And very early on, the hippocampus planning the route, and then lots of other brain areas take over. Right, so let's see if this works. So please take me to Peter Street. There's the hippocampus there, and they're just coasting along. And now, I need to change my mind. There's a quicker kind of way of going. Oh, it's blocked off. So that's, damn, we need to do something else. And then, got to turn around. We see sort of motor areas, cerebellum in the brain. And then there's lots of nice shops down here. You see loads of visual cortex. And we're coasting along. Setting up an expectation. I should see this coming up. Yep, there it is. And a bit of road rage there, just into the bargain. So you can see what we were able to do with this was capture the kind of complex neural choreography of what's going on when you're navigating. And it's not, of course, just to do with the hippocampus, but the hippocampus is playing a really important part. And we know this because we used this very same environment, virtual environment, to test a patient who had been a licensed London taxi driver for 40 years and then had an illness that damaged both of his hippocampi, and he became amnesic. And so the question was, would he still be able to find his way around London that he'd been navigating around for 40 years? So this just shows you the choreography. So here is the patient. You can see his hippocampi here very shriveled. Here is just a tiny part of London. So this is only part of what taxi drivers have to know. And what we asked the taxi driver to do, and these are 10 controlled taxi drivers who are the same age, have the same amount of driving experience as the patient, and they had to go from St. Paul's Cathedral to the Bank of England. And you can see the controls all took their nice quick route. They all knew what they were doing. It's a really common, quite a common shortcut for taxi drivers to take. But the poor patient, he starts off and he is going all around the place. And he cannot get there. And he knows he's wrong. And here he kind of knows he's in the vicinity, but he never gets there. So even after 40 years of driving these streets every day, he had lost this ability. And what was interesting is, if you see, he kind of stays more or less on what we would call the A roads, the main roads in London. So these are particularly well-travelled roads. And he's not able to use the smaller roads. And what he said his problem was, is that he couldn't visualise what was coming next. And he couldn't imagine the route such that he knew where to turn off. And you can see here this exact problem. He should have turned off here, but he couldn't visualise the scene, if you like, of that junction where he was supposed to turn away. So the hippocampus, very important. Even after 40 years, you still need it. Now, more recently, we've been looking at navigation in the 3D world. So everything I've told you so far, and indeed most of the animal work that's done, involves just looking at people on a flat road, a flat 2D plane. But of course, a lot of our activities take place in multi-level buildings. We go up and down the mountains. We often fly in airplanes. We dive underwater. And so the question then is, how do we navigate in these 3D spaces? Because that's much more what we have to face in the world. So we've been looking at this again with virtual reality. We built a kind of a jungle gym for people to climb in. We also had them navigating around virtual buildings with lots of different levels. More recently, we actually had a virtual spaceship, so people were kind of in zero gravity, if you like, to see how they would navigate around. And lo and behold, the hippocampus, once again, was representing places in this 3D world. So it's not just on a 2D plane. Even when you're in a volume like this, where you're floating around, your hippocampus knows where you are. So the question then we are left with is, how on earth is this structure supporting all of this type of spatial representation and navigation, but also somehow supporting our lifetime of memories? So how is it sort of doing these apparently diverse functions? Well, we made the situation even more complicated about 10 years ago, because what we discovered, testing again a set of patients with hippocampal damage, was they not only could they not remember the past, but they also couldn't imagine the future. Indeed, they couldn't imagine any kind of scene or event. So they had a severe imagination problem for scenes and events. So here's an example. Imagine you're lying on a white sandy beach in a beautiful tropical bay. Now the control gives you lots of lovely information about what they're seeing in their mind's eye. The patient, he can't really do this. He can tell you some kinds of items that would be related to this type of scene, but he can't really see it. Can you see anything else? No, it's just like I'm kind of floating. They can't really see the scene. So here we had Another thing that the hippocampus seemed to be important for, not only the past, but also the future, and indeed any kind of imagination task involving scenes. Now, interestingly, these patients were very good at imagining single objects, but they couldn't imagine scenes. And here's just showing you how impaired they were. And here's some examples of, they know they have this problem. So there's no scene in front of me here. It's frustrating because I feel like there should be. I feel like I'm listening to the radio instead of watching it on TV. I'm trying to imagine different things happening. But there's no visual scene opening out in front of me. Or another patient said, it's hard trying to get the space. It keeps getting squashed. So they have some insight into what their problem is. Now, we went on to try and characterize this a bit further in this way. So if I show you this picture for about 15 seconds, and then I take it away, and then I immediately ask you to draw it from memory, you'd think, very simple, I can do that. Give or take a bit of artistic skill. This is what most people would draw. This is from a control, it's not from a patient. And what you notice is all the extra space that was never in this picture is here. And this is only like 15 seconds after they saw it. This effect is known as boundary extension. And it's the propensity that we all have to extrapolate beyond the view in scenes. We do this all the time. It's implicit, we're not aware of it, it's automatic. And it's actually a really good thing to do because even though we're making an error here, it's good to think about what might be around the corner, right? So if you were living in, a jungle somewhere, you would want to know, is there going to be some monster around the corner that's going to eat me up, right? So this kind of extrapolating beyond the view is a very good thing, but it is an error of memory. So let me explain it exactly how it works. So you would see this picture. But what you actually do is you think about what's beyond the view. And you do it instantaneously. So what you actually have in your head is this representation. So when I come along and ask you, is this picture exactly the same picture that you saw at the start? Or is it closer up, or is it further away? Most people say, it's much closer up because they're comparing this to what's in their head. But in fact, it's exactly the same picture. But because you made this kind of extrapolation, you're going to be making an error because most people will say, it's closer up because they're making the comparison here. And the gap between seeing this picture and this picture can be as little as 42 milliseconds. And we make the error. And we all do it. Even children, even babies do this. And it's scene specific. It doesn't happen for objects or anything else. So naturally we thought, well, what happens to patients with hippocampal damage when we ask them to do this task? Because they're not very good at imagining scenes. So this is what we did. We showed them simple pictures, and then briefly, and then we would show it to them again. Always, it was the same picture on every trial. This picture and this picture would be the same. And then they were asked, was it the same picture? Was it closer up or further away? And just like we would have predicted, the controls, the healthy controls, more often made the error and said, oh, it was definitely closer up, definitely. But the patients, the amnesic patients, were much more accurate on this task. They said the same much more often. So here we have a kind of paradox where the patients who are amnesic were doing much better on a memory task than the healthy controls, simply because they were not able to imagine what's beyond the view. So that kind of is more evidence that this scene construction is a very important function of the hippocampus. And here's just to show you some other ways we tested this. Again, here's the pictures shown for 15 seconds. Here's what the controls drew immediately. Again, you can see all this extra kind of space, but the patients were much more accurate on this task. Now, we wanted to take this further, and so we did some more experiments with scenes. So a little bit of audience participation called for now. So if you look at these pictures, can you see anything odd or unusual about them? No? Nothing odd or unusual? Okay, what about these? Anything odd or unusual there? Speak up if you think you know. Say again, chat louder.
Different viewpoints.
Well, these are actually impossible. You could not have these structures in real life, because let's take this one. You see that these pillars seem to be supported at the front, but by the time you get here, they're supported at the back. So these structures couldn't really exist in real life. They're kind of impossible scenes, and a very...
MC Escher.
Exactly, it's very much that kind of idea, and a very similar issue here. What about these ones? Anything odd or unusual here? No? Okay, so here we actually have some sharks in a very tiny pond. And here we have one person who's presumably some kind of vampire, because they're not actually casting a shadow. So what you can see here is this is a different type of impossibility. And this was important because we knew from testing healthy controls, that they use very different strategies to evaluate these scenes. So if we just go back, for these ones, they said what they did was they kind of formed a picture in their heads of what it should look like, and then they compared it to what they were looking at. So this is very much a kind of spatial scene construction in their heads type strategy. They didn't do that for these types of scenes, where they said they just kind of knew something wasn't right and didn't belong in these scenes. So we had these different strategies. So then we thought, well, what would happen with the hippocampal damaged patients? And what you find is that the controls were kind of making similar errors for these semantic, as we called it, the sharks in the pool type pictures, and the constructive errors. But the patients were making lots of errors. They couldn't see the impossibilities in the sort of spatial structural scenes because they couldn't create a model, if you like, of the scene in their mind's eye. But they were absolutely brilliant, so they were almost at ceiling in spotting the semantic errors and spotting the sharks in the pool and so on. So again, this seemed to really underline the fact that scenes and scene construction is a significant problem for patients. And so we're left wondering, well, how does this all fit with the navigation and the autobiographical memories? And so what we did is we came up with this scene construction theory, and the idea that a primary function of the hippocampus is to construct scenes. And any task that requires or would benefit from an internal model of the world or a scene will require the hippocampus. And this isn't just in the context of memory. So memory is one function that certainly benefits from you imagining scenes, but so do other things, like perceiving scenes, like in boundary extension, and also in other aspects of cognition. And this is a very different view of the hippocampus. Up until kind of this point, everybody thought it's a memory device. But what I'm saying here is, it's not a memory device, it's a scene constructing device, and memory happens to be one function that relies heavily on this ability to imagine scenes. But if I'm correct, then as I say here, it should, the ripples should go out much more widely across our cognition. And we've looked at this in many ways, but I'm just going to give you one example of how this works. And it's a form of thinking that's hugely pervasive. We all do it. One estimate has it that we do this for 50% of our waking hours, and that is mind wandering. So we all mind wander a lot. But we wondered, do people with hippocampal damage mind wander? We know they really can't imagine scenes, and I don't know about you, but for me, mind wandering often does involve imagining scenes and what you're going to do after you leave here and all this kind of stuff. So here, again, just to remind you, our healthy controls with our patients, and do they mind wander? And the way we did this is the patients, they come to see us from all over the UK. They're very rare patients. They spend a lot of time with us, 3 or 4 days. And Connie McCormick here, who is leading the study, at just various points where there would be a lull in proceedings, she would turn to them and say, "What are you thinking about right now?" And she would get them to describe their thoughts. And what we found from that is, yes, the patients do mind wander, and they mind wander as much as healthy controls do. So then we wondered, well, what is their mind wandering like? Is it qualitatively the same? Well, it's not really, because what you can see here is these are sort of going into the past, into the future, and the present, that the controls do a lot more mind wandering about the past, while the patients are kind of stuck in the present. They're thinking about the here and now. They're not mind wandering about times past. They also don't mind wander that much in scenes. So the green is how much of the thoughts were mediated by scenes, and in controls, it's a lot of their mind wandering, as I think isn't a surprise to us. But the patients, very little. They just have these kind of verbal thoughts. So what I'm suggesting then is that we have the hippocampus constructing these scenes, and all of these different aspects of cognition require this vital ingredient to really work properly. Now, it's not that the hippocampus is controlling all aspects of these functions, but these functions need this vital ingredient. And, as I said, if you think about it, when you're planning a route, when you're recalling the past, when you're imagining the future, it is very sceney in your mind's eye. So, to conclude then, the hippocampus is not a memory structure per se, it's a scene construction device, I would say, that has a much wider remit across cognition, but is also vital for memory. And this kind of blurs the distinction, really, between the past, the present, and the future, and between, memory and the rest of cognition. And I would say that scenes are the currency of cognition, and they kind of oil the wheels. And when that ability is diminished in the context of hippocampal damage, then the effects can be far-reaching and really quite devastating. So, just for the end of the talk now, I'm going to consider, well, where do we go from here? Well, the hippocampus, it turns out, is not a uniform structure. It's made up of many different parts, which are known as subfields. And we're able now to really get a good handle on these different parts of the hippocampus using high-resolution MRI scanning. So we can see the different subfields, and now we can start to understand how information flows around these different parts of the hippocampus, how they talk to each other, how they talk to the rest of the brain. And so this is going to be very important for the future to understand the mechanisms that are helping us build scenes and remember things. We also have much more powerful MRI scanners now. The magnets are much more powerful. So for example, in our current scanners, which are 3 Tesla scanners, this is kind of the size of the voxel that we can scan. So it's about 1.5 to 3 millimeters, which is pretty good, you might think. But with these new scanners, a 7 Tesla scanner, we can now scan even more detail. So we can see the brain in ever finer detail, and we can understand how the information processing is done, and of course, how it goes wrong for the patients. We also have quite sophisticated techniques now for understanding how the hippocampus and its various subfields talk to the rest of the brain, and what areas are driving other areas, and so on. Now, this, as I showed you, was the standard MEG scanner, which gives us this beautiful millisecond precision. But you can see that it's very restricted. This guy ain't going nowhere. He's sat there looking at this screen. But we have developed a mobile MEG scanner in our centre, and it uses something called optically pumped magnetometers. But basically, the bottom line is people can now move around, they can be tested for hours at a time, and we can record neural signatures with still the millisecond precision, including areas like the hippocampus. Now, we've never been able to do anything like this before. Here's what the original prototype looked like. So we have our friend here. He's got this kind of helmet on, and in the slots fit these OPM sensors. So they're actually resting right on the head, which means we get brilliant signal. And he's also, believe it or not, even with all this wiring, able to move his head around. These are the next generation, which we're about to take delivery of. Much smaller, much neater, much more user-friendly. Now, my colleagues who developed this have just reported on the sort of proof of principle of this in Nature. And so this is Elena Botto, and she's actually doing a finger tapping task, which is kind of classic, activates the motor areas of the brain. But while she's doing that with her left hand, she's drinking a cup of tea with her right hand, and she's moving her head, and she's doing all of this. In another experiment, again, she's finger tapping here, but she's playing ping pong. So she's trying to do as much of this, so she's doing lots of moving around. And yet, we can still get these beautiful focal activations in motor cortex using this technology. And so what we're planning to do now is to move on from the ping pong, and we've had people constructing scenes using this mobile device. So they're sitting there and they're going, well, I would do this, I can see this or that, and we get beautiful activity changes in the hippocampus. This is just showing you 3 subjects here. But of course, we always want to push it further. And so what we're going to do now is take advantage of our experience in virtual reality, and we're going to combine this immersive, interactive virtual reality with the mobile brain scanner. We're even going to have people on omnidirectional treadmills, so they'll be able to walk through virtual worlds, all while we create experiences for them to have, and we'll be able to capture the formation of memories and then track what happens to them over time subsequently, and when they go awry in patients. So this is kind of cool new technology. We've never had this capability before, so it's quite exciting. And I'll just end by noting that, of course, it's also a kind of boom time for artificial intelligence. So Demis Hassabis was my PhD student, and he's got a company that's been acquired by Google, and they've been doing amazing jobs teaching computers how to be very clever. So you might have heard about this. This is the game Go. It's a very tough game, and until now they really couldn't get computers to play it very well. It's much more difficult than chess. And this is Lee Sedol, and he was the world champion at Go. Really phenomenally good player. And Google DeepMind built AlphaGo, which uses these deep learning algorithms, and it was able to teach itself how to play Go. and in the end actually beat the best human at this. So artificial intelligence is also becoming ever more sophisticated, but interestingly, how Dennis and his colleagues got this to work was they looked at how the human brain operated, and they took a lot of neuroscience knowledge and started to apply it to artificial intelligence. And then you can see that you can come up with some cool results like this. So I think, as I said at the start, there's still lots we don't know about how memory works, but it's an incredibly exciting time to be working at memory, and I think we are on the road, I would say, to cracking the memory code. So thank you once again, and again, thank you for the invitation from the committee to Lethbridge. Thank you.
Okay, so we've got some time for questions. If anyone has a question, you can raise your hand. You're going to have to shout at me though, because Eleanor is going to want to use the mic.
The study of the mouse